Have you ever stopped to think about how life actually works? How a single, invisible line of code can build everything from a microbe to a blue whale? Well, today, we're diving into that story. This is the ultimate instruction manual for life itself.
Just think about this for a second. You've got trillions of cells, right? And inside every single one of them is the complete, unabridged instruction manual for building you. Everything is in there, from your hair color to how you digest your lunch. It's all written down in a master blueprint. Okay, but here's the catch. This blueprint, your DNA, is so ridiculously important that it's kept under lock and key in a vault we call the nucleus, and it can never, ever leave. So if the master plan is stuck in the head office, how does any actual construction get done? How does the work on the ground floor even start?
The answer to that puzzle is something with a kind of intimidating name, the central dogma of molecular biology. But don't let the name fool you. It's not really a dogma. It's more like a workflow, a beautiful, efficient system that describes how information flows from that locked vault out to the rest of the cell.
The easiest way to get it is to think of it like a giant construction project. So you've got your master blueprint, that's the DNA, tucked away safely in the main office, the nucleus. Now, when you need to build something specific, you don't take the whole blueprint out, right? Of course not. You just make a quick photocopy of the one page you need. That copying process, that's called transcription. Then that copy, which we call mRNA, gets sent out to the job site. And there, the construction crew, a ribosome, reads the copy and starts building the skyscraper, which for a cell is a protein. It's that simple. Office, copy, job site, build. All right, so let's zoom in on step one. Let's talk about that master blueprint, the DNA. Your DNA is, quite literally, the library of you. It has the instructions for everything your body will ever need to do. And because it's so precious, it's locked away in the nucleus, where nothing can mess with it. Any damage to the original could be, catastrophic. That's also why, before a cell splits in two, it has to make a perfect, flawless copy of the entire library. We call that replication. It's all about protecting the source code at all cost.
Okay, so if the DNA can't leave the office, how do we get those instructions out? This brings us to that crucial photocopying step.
This is where a process called transcription comes in. You can imagine this amazing little enzyme called RNA polymerase as a super advanced portable scanner. It just glides along the DNA, finds the exact gene, the one page of the blueprint it needs, and makes a quick, disposable copy. That copy is called messenger RNA, or mRNA for short. It's the perfect little courier, designed to carry the message out of the nucleus and into the wild.
But wait, in cells like ours, that message isn't quite ready to go yet. It has to get prepped for the journey. Think of it like putting on armor. First, it gets a special protective helmet called a 5' cap. Then it gets a long, stabilizing tail, which helps it last longer. And finally, in a really clever step called splicing, all the gibberish parts, the introns, are snipped out. So all that's left are the actual, useful instructions. Now our messenger is ready for action. The message has been sent, it's been prepped, and it has arrived at the job site. Now it's finally time to start building something. And that job site is the ribosome. This thing is an absolute marvel. It's the cell's protein factory. In our analogy, this is the bustling construction site where the blueprints are about to be turned into a real physical thing. This is where the magic happens. And this factory runs like a well-oiled machine. It's basically a 3-station assembly line. A, P, and E. Simple. At the A site, A for arrival, a little delivery truck called a tRNA shows up carrying a single building block, an amino acid. At the P site, P for peptide, that new block gets slapped onto the growing protein chain. And finally, at the E site, E for exit, the empty delivery truck takes off to go grab another block. It just keeps going in this rapid-fire cycle. Arrive, attach, exit, over and over again. So we see the factory line, but how does the ribosome actually read the instructions? What language is this all written in? Well, the genetic code has some pretty simple rules. It's read in 3-letter words called codons. Each 3-letter word stands for one specific amino acid, one type of brick. The word AUG is basically the universal signal for start here. And then there are 3 words, UAA, UAG, and UGA, that mean, okay, stop.
What's really cool is that the code is degenerate. That just means that a few different words can stand for the same brick. It's like having synonyms, which is actually a really clever feature.
And that redundancy is explained by something called the wobble hypothesis. It's a fantastic idea. It basically says that for a lot of these three-letter words, only the first two letters have to be a perfect match. That third letter can wobble a bit. And this is a huge deal for efficiency. It means one delivery truck can recognize a few different but similar instructions. It saves the cell from having to make a unique truck for every single possible word. And speaking of saving energy, building stuff is hard work. It's incredibly expensive, energetically speaking. How expensive? Well, take a look at this number. To add one single amino acid, just one brick in the wall, it costs the cell the energy from 4 high-energy molecules. Your body is doing this millions of times a second without you even thinking about it. It's absolutely mind-boggling.
So, why have we gone through all of this? Why does understanding this system in such detail actually matter? Well, because once you understand how a machine works, you also know how to break it. And that is the secret behind so much of modern medicine.
Just look at this. A ton of our most effective antibiotics are just expert saboteurs. They work by jamming the gears of this exact process, but specifically in bacteria. Rifampicin, for instance, blocks that scanner, RNA polymerase, so the bacteria can't even make copies of its blueprints. Others, like tetracycline and chloramphenicol, mess with the ribosome factory, stopping the assembly line cold.
The absolute genius of this is how specific it is. See, the construction equipment in bacteria is just a little bit different from our own, and that difference is everything. It lets us design these drugs to be like molecular sharpshooters. They go in, shut down the bacterial factories, and leave our own cells completely alone. It's an entire field of medicine built on knowing this fundamental process.
So there you have it. We've gone from a locked blueprint to a temporary copy to a finished protein that does a job. We've even seen how we can hack the system to fight disease. Understanding this process has given us the power to read, write, and even edit the code of life itself, which leaves us with one heck of a question. Now that we understand the instruction manual, what are we going to build next?